Oh,
Hello there. I'm Mr. DNA. You might know me from classics like the Jurassic Park introduction video. And that's it. It didn't go so well. Anyways, today I've been asked by the suits over at Jurassic World to explain exactly what happens to DNA as we duplicate it. Let's start with the basics. Me. This is my father, Mr. DNA Sr. Like all DNA, he is made up of the four nitrogenous basics and a helix of sugar phosphates. These nitrogenous bases, along with the sugar and phosphate, are in sets called nucleotide bonds. These bonds serve as information that facilitates the creation of proteins through specific codes.
DNA is separated by a protein enzyme called helicase. Helicase separates the double helix strands at the hydrogen bonds between the nitrogenous bases. Free bases are then attached to the split strands of nitrogenous bases using a complementary pair. Remember, adenine binds to thymine, cytosine binds to guanine, but because the free bases are forming mRNA, adenine would bind with uracil. This process is called transcription. And now that the mRNA is copied, it can be translated. The complementary mRNA formed by the free bases then detaches itself from the DNA strand and is released through the nucleus into the cytoplasm.
eventually, the mRNA finds a ribosome. These ribosomes read the specific coding within each mRNA through codons. A codon is a 3-code sequence to determine which type of amino acid is formed. These codons serve as a code for the ribosome, who creates the long chains of amino acids. These amino acid chains are also more commonly known as proteins. This process of translation happens a massive amount of times. Okay, boys and girls, now that you know how DNA is translated and transcribed to proteins, if you're lucky and you keep learning, you'll work for Jurassic World. I'm sure nothing will go wrong. Bye now.